Hi, everybody, and welcome to our lecture today, where we are going to focus on the concept of um, kind of complex inheritance, moving away from the Mendelian genetics, where we talk about one gene affecting the population. So for this one, you guys are going to learn a lot of new vocabulary and then explaining those vocabulary in terms of how it affects the population and passing the uh, genotype and phenotype to the offspring. So as we talked about Mendelian ratios in our previous lecture, we can use our Punnett square to figure out how a single gene can cause a specific characteristics in the human or in terms of development of certain disease or a person being a carrier versus having the actual medical condition. Now, most genes that we are dealing with in humans are not considered to be simple inheritance following the Mendelian genetics, rather than they are more complex. However, this is interesting to note that there are three general phenomena that seem to be the exceptions of the Mendelian law. However, in reality, they're not. This is related to the gene expression, which is a focus of our lecture today, inheritance through mitochondrial DNA, as well as the concept of linkage. And again, we're kind of connecting those to the Mendelian ratio and how does that affect it? How is it being affected if you have number of genes influencing the final outcome of the genotype and phenotype? So one of the first vocabulary we want to learn about is the word lethal alleles. So as the name word lethal implies, it means if you have a certain combination of the alleles being passed on to the offspring, that leads to the offspring being killed off or unable to reproduce as a result of dying shortly after birth. So lethal genotype, the best example that we have is when you have a homozygous allele being present, and the homozygous allele literally causes such a medical condition that either literally kills the individual or kills the offspring, or causing it to basically a very high overproduction of certain situations or certain production of overproduction of specific proteins that again leads to death, hence the word lethal. So in this case, if you are carrying a homozygous dominant version of the gene, this is considered to be a lethal allele and therefore leads to the death of that individual. However, if you are homozygous recessive, you will consider to be a normal person, and if you are heterozygous, you are a carrier, you do have the medical condition, and however, you are surviving the environment and therefore can pass that genetic information to your child. Now, in this case, the example we're using is a medical condition called achondroplasia, which has to do with the height of the individual. Again, if you consider an individual having two copies of the dominant version of his gene, this person will not survive and will die shortly after birth. Um, if they are homozygous recessive for this characteristic, they're considered to be at a normal height. And if you have the heterozygous characteristic, then you would basically cure the symptoms of this disease or this medical condition. For hyperplasia, this is basically the height is not as tall as a normal individual and or normal height of an individual and in symptoms. Now again, this is a double dominant, so as you can see, it's capital A, and that's where you actually will see uh, either the overexpression or malfunctioning of um, the um, the genotype, and therefore that leads to the lethality. Here is a really nice Punnett square that highlights how an individual's offspring may be influenced if they are both carriers, as well as they have the medical condition. So here we have the two parents that are heterozygous for the medical condition, which means they have the um, uh, shorter height, and we want to see using the Punnett square, how does that bag is passed on to the next generation. So you have the genetic information of each parent, so the genotype of each parent. You have this individual, which is going to be basically, part of the person that it would be the individual that would be dying off as a result of the lethal alleles. This individual right here will have the normal height because it's the um, homozygous recessive version, and these two boxes are going to be your chance of you having a heterozygous, um, where your height would be lower, you are the carrier of the medical condition, but you would survive the medical condition, and it doesn't consider lethal. One thing I do want to note is notice that the numbers that we have here are divided by 3, not 4, because we're assuming that the child that has the homozygous dominant gene, which is in red, will pass away, which means we will only looking at a 3 possibility of that embryo. So down here it says, if two people with achondroplasia have a child, we're considering this again 2/3 for the heterozygous and then 1/3 for the homozygous normal height. And this is assuming that the last child will not survive. You may also take a look at if you have a person who has a normal height, so homozygous recessive, kind of marrying somebody or having a child, I should say, not marrying, having somebody with achondroplasia, what is the chance of their child being normal or having the achondroplasia? And you notice if you follow the Punnett square, you have 50% chance of having a normal height and then 50% chance of having the achondroplasia or achondroplasia. Now, multiple alleles, as the name implies, it's pretty self-explanatory. It is a situation where you have more than 2 typical alleles that we have. We learn about the alleles being kind of the dominant versus the recessive. But in this case, we're talking about multiple alleles affecting or having 4 different type of alleles. So we have the dominant and the recessive version, the homozygous, and then we have 2 additional genotype, which is lowercase c, with this variation with the CH, and then another variation with H and H on the top. So we have total of 4 alleles versus a typical 2 alleles. And as a result, we can see that we have 4 distinct coloring related to these alleles for these rabbits. Now, the concept of incomplete dominance, again, if you kind of put your mind into it, you can use the terminology to define it. So as the word incomplete implies, this is going to be related to where you have a heterozygous phenotype being reviewed. And because it's incomplete, you neither have the characteristic of the homozygous dominant nor the homozygous recessive. So you fall somewhere in between the two. So if you are crossbreeding a true breed of red with a true breed of white, which are the 2 homozygous, you will end up with 100% heterozygous genotype, and the phenotype of that would be a pink color, which is again kind of lies between the 2 of the dominance characteristics. Another example of the incomplete dominance is related to a medical condition that lead to an elevation of cholesterol in a bloodstream. It's known as familial hypercholesterolemia. Now, again, we have the 3 scenarios where you have the homozygous dominant, you're identified as a normal individual. You have the correct number of receptors, where allows the bad cholesterol or LDL cholesterol to be attached, and therefore bring into the cell. And this is going to cause a low level of cholesterol in your bloodstream, which is what we typically want. And this is the general population. You want kind of a number under 300 or 200 for a typical population. Now, again, if you have a homozygous recessive, you will have zero receptors on the surface of the cell, which means your cholesterol is going to be super elevated. So cholesterol is going to be super high, and you can see these are the individuals that are basically the individuals with a medical condition, which is the familial hypertension. Now, heterozygous individuals will still have the condition of elevated cholesterol level, which is the picture you see right here in the middle. They do have receptors, but the receptor frequency is about 50%, which means they have an elevation in the cholesterol. However, the elevation is below what's considered as a homozygous. So this is what you have. So it's somewhere between the people that are healthy and some people who have the full-blown medical condition. Another example of another condition related to the genotype and the phenotype is the concept of codominance. I think about codominance as two things being happening at the same time. So when you have a heterozygous phenotype, both of the genes are being expressed. So both of the alleles are going to be expressed versus one being a dominant allele over another one. And the best example of the codominance that we have would be a sample of blood typing. So for this, we have the two main alleles, which is IA and IB, that produces antigen A, antigen B on the surface of our red blood cells, as you can see right here. And if you have the allele that is lowercase I, this is an allele that does not produce an antigen. So this is again, you have multiple alleles rather than the typical. Now, if you have both IA and IB alleles present in your genotype, in this case, both of them are going to be presented on the surface of the red blood cell, making your blood type AB. However, it is to be noted that both of the IA and IB are considered to be fully dominant over the IA, which is the so here is a depiction of the main blood type. If you have 1A or 2A, you have L. If you have the heterozygous B or a homozygous B, that's how it is. And if you're heterozygous or A, that's how it is. Both of the alleles will be presented with one that's not overshadowed the other one. And if you have allele II, then you have no antigen present, which makes your blood type an O blood type. Now, expanding on the same concept of blood type, a new concept also comes into play, which is called epistasis. Epistasis is where you have one gene affecting the expression of another gene. So you have the involvement of two genes when one is the expression of one gene being controlled by a secondary gene. So Bombay phenotype is an example of the epistasis. So H gene, so capital H gene is the epistatic gene to the I gene. So what does that mean? This is really actually interesting. So if you have the H gene, okay, so if you have the H gene, you produce the H protein, which means that you can have a red blood cell that has the antigens, so this is like your little H, and then the A and B can attach to them. So the H, kind of think about it as the anchor for the A and B antigens to attach on the RBC surface or red blood cell surface. So if you have either the heterozygous or the homozygous dominant version of this gene, the H gene, your blood type will be any combination of the codominants we talked about, either A, B, or AB. However, if you have the homozygous recessive version, which is lowercase h, you will not have the H protein present, which means you have no anchor. So even though if you have A and B, right, if you have your A and B, you cannot attach them. So it means that your blood type will end up with an O, even though you have the allele A and B present, but because the H is in the homozygous recessive version, that genotype of A and B will not be expressed. So that's very cool because, again, the H is kind of controlling how your body represents the allele A and B for your blood type. Now, phase is another good one. Phase is where you have one gene and damage or I guess changes in that gene can actually affect multiple systems and multiple parts of the body at the same time and lead to multiple disease. For instance, if we have a mutation related to the beta globin of the hemoglobin, causes the sickle cell anemia, not seeing the damage to your red blood cells is a typical thing we see. We also see sickle cell anemia may affect the features of the nervous system, affecting and causing blindness. It may cause liver failure, so it's affecting other organs and organ systems, and it may have a higher chance of developing heart attack. It's affecting your cardiovascular system. Again, even though you're only talking about one gene, it has a very high reach on the rest of the body and therefore affecting multiple systems in the body. Another example of this is called Marfan syndrome, which is a medical condition where a specific elastic connective tissue called fibrillin is not forming properly. And in a normal form, this is what it looks like to be. It forms beautiful linkage together, making this elastic fiber very strong. However, if you do have an alteration in the corresponding gene, your fibrins are too little, they're more curved, and therefore, when they form together, the linkage between them are very weak. Now, even though this is just an elastic protein, it actually affects major parts of the body. One of the parts that are affected is your joints, making you much more susceptible to dislocation. You're also going to have longer limbs, spindly fingers, and one of the worst things is affecting the development of the thoracic cavity, causing a caving chest. So basically, the chest cavity will be smaller because the rib cage is not fully grown. Again, most of these symptoms are very dangerous for the individual, but even worse than that is that lack of this elastic fiber is going to affect the aorta because aorta has a lot of elastic fibers that gives it its flexibility, and therefore these individuals are going to be susceptible to having their literally aorta bursting open as a result of absence of these elastic fibers. So genetic heterogeneity, I can't pronounce it now. Geneity is when you have multiple genes in your body causing the same medical condition or disease. Basically, a same phenotype comes out as the end of that process. For instance, if you have an enzyme if you have a pathway, biomedical pathway, which involves, let's say, 10 enzymes, right? If any of these enzymes in this pathway being mutated or changed, the final effect will be shown in that pathway. So it will affect the overall pathway regardless of which of these genes and the protein in the pathway going to be changed. So basically, if you have one protein leading to action of the second one, leading to action of the fourth, and then fifth, and etc. If this is your pathway, and if there is a problem in this gene, the final effect will be same, right? If one is healthy but two is affected, again, the final product will be same or affected. Same thing with 3, 4, and 5. So you can have all healthy genes in a row with the exception of one of these, and the final effect will always be the same. So example of these medical condition or this situation where you have multiple genes causing the same outcome is osteogenesis imperfecta, where 8 different types of mutation in the gene have the same effect on the body, causing the osteogenesis imperfecta, which is basically a problem with the development of the skeletal system. Another one is retinal dystrophy, which is again causing mutation in 20 genes. So in the example that I gave you, I only had 5 genes, but if you have mutation in any of these 20 genes that basically help you see, that function will be affected. Hence, you will cause blindness. So examples of some of these genes that you have listed. Again, mutation of any of these genes into the of damage to the photoreceptors of the eye and therefore affecting the image created by your eye, leading to blindness. Now, another thing that we talked about was the mitochondria. Now, we understand that our cells have a nucleus, and within that nucleus, we have our genetic information stored in form of DNA. And then anytime we want to make proteins, we make copies of the corresponding genes and having them expressed using our ribosome. Now, mitochondria, which is this double membrane organelle inside our cell, is very unique in terms of the function and in terms of its features. So its function is extremely important because it is responsible for production of energy in a form of ATP for your cell. So your cell basically depends on the mitochondria to survive. It kind of makes sense. Now, the other component is that mitochondria has its own genetic information in form of these little circular DNA called mitochondrial DNA or mtDNA. It's a combination of about 37 genes that have been only inherited from the mother to the child. In terms of, at the adopted child to see who their biological mom is, you can look at the mitochondrial DNA. It is important to understand that this inheritance pattern from the mitochondrial DNA versus the regular genes is different, and the mitochondrial DNA is more susceptible to mutations. Now, as I mentioned, mitochondrial DNA does get passed down from the mother. So in this case, you have the black representing mitochondrial DNA, and you can see each of the child, regardless of the male or female, are. And the next one, again, if you have a girl, that girl is also going to have this two braces. However, if you have a son that is represented with the black square, that's going to be. Now, because the cells typically have thousands of mitochondria, each of them have a number of these mini chromosomes inside the mitochondria. As you can see, kind of like these little loops here. Okay? And again, it's very important for us. This mitochondria is very important for us because it's the energy source of your cell, right? So they're the ATP product. one of this is called mitochondrial myopathy, which is associated with weakening of the muscle, and then liver optical atrophy, which causes damage to the uh, retina of the eye and causing inside vision. Even though mitochondria conditions for you. And theoretically, if you do have um, some sort of disease and you don't want to pass, so if you're a woman and you have certain medical conditions through the mitochondrial DNA and you do not want to pass this to your kids, you could have the mitochondria being replaced by a mitochondria coming from a donor. And as a result, make sure that your child does not carry the diseased gene. Now, the last concept we're going to talk about complex inheritance is the concept of linkage. And linkage is pretty straightforward. I think we're going to try to keep it as simple as possible. Now, we need to understand that when you have genes that are relatively close to each other, they're considered to be linked. As you can see on the top of the image, we have the two genes P and L. You can see that they are within proximity of each other, and therefore, wherever there is a close proximity of these genes, they will not be independently assorted, which means anywhere the P going to go, L is going to follow. Or anytime the L is going to go, the P is going to follow. So there is no, if there is a linkage, or if you have what link, they do not separate, they do not undergo crossover, which means they are not independently assorted during the process of meiosis. Now, linkage genes, as I showed you here, I think it's very important to you guys to note this. So on the first image you have, or part A, you have genes that are literally on the opposite side of the chromosomes and two different chromosomes. So let's say if this is my chromosome 1 and this is chromosome 2, these P and L are not only on two different chromosomes, they're also in the opposite arms of the chromosome. And therefore, they are going to be independently sorted and passed on to the next generation. So we can use a typical Mendelian ratio to solve for this. Again, the concept is because they are unlinked, they are independently assorted, which means they are very easy to figure out using the Punnett square to figure out the frequency of each gene. However, when we are looking at linked genes, like the PL genes on the second example, you will have no independent assortment, which means basically these two genes are always going to be together and these two are going to be together. So when we write them together and we create one pattern for them. So it's always capital P, capital L, lowercase p, lowercase l, versus no, you can have this one crossing with this one crossing with this crossing with this, and this crossing with this, which gives you these four options right here. Now, in this case, notice we only have two options because there is no independent assortment, which means P and L are always going to stick together. Now, recombination is a result of the crossing over. Now, again, this is going back to the concept of linkage and if the linkage is present or not. And if it is present, then there's a chance that they are going to be carried through the recombination DNA together versus independently. So chromosomes recombine during which phase? They recombine during the phase called prophase 1 of the process meiosis, which forms your gametes. And the parental chromosomes will have the original configuration, which we already know. And then recombinant chromosome is after you have done your crossing over and you have created a new combination, hence the word recombination. As in you kind of like shuffle things and then you reorganize the alleles based on that. Now, we can use this concept of recombination to figure out the linkage map. Are these proteins linked together? Sorry, I should say protein. Are these specific genes linked together? And again, or are they separated from each other and therefore can undergo recombination and independent assortment? So correlation between crossing over frequency and the gene distance is very important for us. Again, if the genes are close to each other, there is always going to be recombination at the same time together, versus if something is far apart from each other, there might be crossing over that are completely unrelated. So, for instance, if you look at the genes A, B, and C, A and B are very far apart from each other and therefore can complete the process of crossing over and basically carry the I should just say leave it at this. They cross over, there is a chance of recombination. But B and C, on the other hand, are very close together. They're linked protein, right? Which means the chance of them crossing over would be less likely because again they are very close in proximity together. So if it is to happen, they will go together and not separately. We also have a situation where we can use the word cis configuration versus trans. So anytime you hear the word cis means that they are on the same versus trans on the opposite. So in this case, the word cis implies the two dominant or two recessive alleles are in each chromosome. So the first example is a cis. Notice this one has the dominant and this one has the recessive version of these linked genes, versus the trans version has the combination of one of the genes being recessive and the other gene being the dominant gene. So that's literally what the meaning of the cis versus trans is. Now, again, kind of thinking about the linkage and the fact that if the two genes are very close together, they do not independently assort. However, if they are far from each other, there is a chance of independent assortment. So when two genes are widely separated on a chromosome, so again, like one end to the other end, so there is no linkage, right? There is a very high likelihood of crossing over, which means that you can kind of move things around a little bit during the meiosis, prophase 1 of meiosis, and you have a high chance of independent assortment. So let's take a look. In this example, A and B are considered to be, I think it's a little bit backward. So this is linked genes and this is unlinked genes. So this is, oh no, sorry. My apologies. This is the linked and this is the unlinked. So there is a crossing over. In this case, the parental allele conformation does happen. In this case, they are in two separate chromosomes, which means that the chance of them undergoing crossing over, not crossing over, sorry, independent assortment is pretty high, and therefore forming the parental genome have a more frequent option. But here, if you have linked genes, you have a higher chance of doing the crossing over. If you have non-linked genes, you have a chance of completing the independent assortment. The idea here is that in cases, it is hard to distinguish what is a crossing over example and what is an example of independent assortment. Because at the end of the day, they both create similar results. So the idea here is if you have two genes that are widely separated on a chromosome, the chances are that those two genes are undergoing some sort of crossing over, but they are from the same chromosome, right? So chromosome 1 and chromosome 1. However, if you're looking at genes that are not linked, let's say on chromosome 1 and chromosome 2, they may create similar results through the process of independent assortment, but they will look very similar to the recombinant gene as a result of crossing over. So do you need to be able to distinguish them? No, but I do want you to understand that the result created as a result of crossing over and independent assortment may look similar, but it's the difference of are they on the same chromosome versus on different chromosome? Because if they're on different chromosome, it's a non-linked gene, which means it's talking about independent assortment versus they're on the same chromosome, that we're talking about the crossing over. Now I'm going to finish with this topic, which is the linkage map. We can use the distances of each gene on the chromosome to figure out how they have lined up. So basically mapping your chromosome. So for instance, if I say the X and Y distance is about 10 cm, which is centigram. There you go. And then X and Z distance is 4 and Y and Z is 6. I can figure out that X and Y are going to be the ends, right? Because that's the longest one. And then Z is going to be in the middle because it's overlapping. Hence, this is going to go in the Z. And then X is on one end and Y is on the other end. So you have 4 and 6, and the combination of the two smallest ones should be equal to the larger one. And again, these numbering and the spacing between the genes is literally helping us figure out the order of the genes and if these chromosomes are actually linked together based on their proximity or not. So again, for this lecture, guys, please make sure you focus on the vocabulary and being able to distinguish what is considered to be a codominance, what is considered to be incomplete dominance, examples of each. So if I provide you an example, can you provide me a reasoning why you have chose or selected a specific type of recombination for the example. And then make sure you understand the concept related to the mitochondrial DNA, how it gets passed on, who passed it on, what's the chance of having mutations in it, and then also linkage and linkage map should be your final goal. Let me know if you guys have any questions, and best of luck.